Welcome to Basic Mosquito Biology. Before we begin learning about morphology, let's talk a little about IPM structure and practices. As you can see, the simplified IPM, or Integrated Pest Management structure, starts with education and ends with pesticides. This is contradictory to the treat-first mentality adopted by the general public. As you can see here, the IPM process can vary in complexity based on impacts from various sources such as the environment, available controls, cultural habits, and behaviors, and suspected population sizes. Considering these factors amplifies IPM structural complexity even further and helps your municipality determine mosquito impacts, treatment thresholds, and targeted control approaches. In order to implement or even understand an adopted IPM, several key things must be learned first. One, must learn to accurately identify local mosquito species within various life stages. Habitat types must also be learned to help narrow down species type and create effective management methods. And one must also have a complete understanding of the mosquito life cycle. With all that said, let's briefly go over each basic IPM component. We're going to start with education and outreach. This is usually in the form of some community event or outreach program. Attending events and or simply engaging residents about their individual cases and or needs goes a long way to helping combat mosquito problems on private property. Next, we're going to talk about surveillance and monitoring. This is usually one of the main jobs of any municipality mosquito program. It allows for consistent and constant analysis of population growth and density within localized regions. This can also be a precursor to determining pathogen transmission when the data is analyzed. Surveillance is usually conducted through the use of various types of traps. Some of the more common ones include the ones that you'll see in the following slides, starting here. The gravid trap tends to focus on Culex species mosquitoes and trapping them as they're looking for sites to lay eggs along the waterline. The BG sentinel trap, which mimics mosquito hosts through lures, traps various mosquito species types that are looking for blood meals so that they can then generate eggs. The CDC light trap, which also mimics host, is similar to the BG trap in that it attracts hungry mosquito females. Another IPM practice is sanitation, or the practices of managing mosquitoes through the elimination of egg-laying habitats. As seen on this slide, there are numerous usable habitats, and some examples will be seen in the follow-up slides as well. Here you have a corrugated pipe which has water resting in each of the corrugations, which provides ample habitat for various species of mosquitoes. Most commonly, however, we see that Aedes albopictus tend to utilize these particular habitats more often than other species. Additionally, you'll see things like tarps and boxes, buckets, flower pots, etc., laying on people's properties, all of which contain small to large size pools of water, which can be very important for resident mosquito populations as developmental and growth habitats, as well as egg-laying sites. Once IPM practitioners have identified populations through surveillance, education, and helped with sanitation determinations, it may be time to implement one or a combination of the many types of control techniques listed here. Which control technique do you think your municipality uses and why? While I won't get into all the specifics, it is important to know that while similar, natural and cultural controls are different in that cultural control is achieved with human intervention, even though the method used may be natural. For example, they may be using a predator that is naturally a mosquito predator. However, it's been transported from one area to the next area, which means that humans have intervened in the process to encourage the predation. Therefore, it would be distinctly different from the other methods of control. After control is selected, it must be applied as seen here and can include one of these various methods that you see in the pictures in the background. So for biological control, up in the top left-hand corner, we have the gambusia, all right, which can be a clear example. Toxorynchides can be a good natural example. Mechanical controls can be various types of traps set, and more commonly, window screens, air conditioning, fans, nets, things of that nature are considered mechanical controls. And then you have cultural controls, which are any type of control methodology that disrupts the relationship between the pest and the environment. So it can be something as simple as emptying out your bird bath. Various types of control can also consist of genetic controls, and while genetic control is effective, it is often too complex and expensive for regional and or widespread use. Therefore, most municipalities will not be using applied genetic control within their management plans. Finally, we get to what is more commonly thought of when it comes to management of mosquitoes and insect pests in general, and that's chemical control. Chemical control should be used as a last resort or as an aid to other methods which are used during the IPM processes. Now, with the IPM having been discussed, let's move to the biology and identification practices that are necessary. Basic biology should always start with a complete understanding of the mosquito life cycle. The basic life cycle structure is egg, larva, pupa, and adult. But as seen, there are 4 substages of larval growth highlighted by a molt. These are known as larval instar stages and can help you when it comes to determining basic mosquito identification. For example, it would be easier for somebody to observe a larva, in the 4th instar stage when they're looking to do identification. And that's just simply because it's so much bigger and easier to observe than, say, an organism in the 1st instar stage. So what else should be considered? Stage 1 of consideration should be looking at the eggs. Various groups of mosquitoes have specialized egg deployment tools or capabilities. Right? As we see here, the Aedes mosquito can often be identified by singular eggs laid in areas of expected water saturation, which also means that if there's an area where they believe that water will be at some point in the future, that's typically where they'll try to lay an egg. The example is the side of a flower pot. So this means that the area of deployment can be dry and the egg structure will protect it from desiccation. Anopheles mosquito eggs, while laid singularly, have attached floats which help distinguish them from the Aedes species eggs, which are also laid singularly. The presence of the floats also means that eggs are often laid on the water surface or on moist soil. Very rarely, if at all, will you ever find any laid on a dried surface. If so, it's oftentimes considered a mistake. Finally, Culex eggs are highly recognizable because they are clustered in rafts where eggs are glued together and then laid on the surface of water. There are also various ways to identify mosquito larvae. The simplest and most common way to narrow down groups is to observe larval orientation at the water surface. As seen in this slide, here, Anopheles is parallel, while Aedes and Culex are both resting at slight angles. So that's a very good way to distinguish between groups when going to look for genus. While potentially being the easiest life stage to identify, species identification at the pupal stage can be quite complex. However, through observation, it can be seen that Anopheles mosquito pupa have a high body surface to water surface contact rate, while Aedes and Culex have fewer points of contact. However, if in the pupal stage, it may be advantageous to place specimens in an emergence chamber when attempting to ID. That way, in a few days, when they come out, it'll become an adult, and it'll be a heck of a lot easier for you guys to identify. There are some basic identifiers which become present after the adults emerge too. First is the palpal length, as seen in the lower left diagram on these pages. Palpal length of the Anopheles mosquito species is generally very long, so it's going to be highly distinguished from the other two categories of Aedes and Culex mosquitoes. And in addition to palpal length, you may also look at the angle at which mosquitoes tend to rest. Resting orientation is another way to help with genus categorization. All right, as typical of all insects, mosquitoes have bodies that are segmented into three sections: the head, the thorax, and the abdomen. The head contains most of the sensory apparatus, such as the eyes, the antenna, the palps, the mouth parts, things of that nature. The thorax usually has all the muscular structure, so therefore we start talking or associating with movement such as the legs and the wings. And then we have the abdomen, which does most of the body processes such as digestion, excretion, and reproduction. All right? Each is responsible for their own specialized section functionality. On the head, special attention is frequently paid to the palps, as mentioned before, specifically the length and orientation. However, occasionally with some groups or species, you may be referencing it for banding as well, which is the coloration or distinctive coloration differences of the scales on those palps. You may also be looking at the proboscis. Okay, the shape here is distinctive, as you can see with the Toxorhynchides. It has a curvature that goes down, or you may have species which also have banding of scales across or in larger sections on the proboscis as well. On the thorax, special attention should be paid to the scutellum, or the thorax shield, as it's known in some cases, where scale size, placement, number, and color can help with identification, particularly if you're using a guide or if you're using a dichotomous key. Knowing the placements of these things and what they are, it becomes even more important. Attention should also be paid to the tiny hairs or setae. Their placement, their size, where they are grouped tends to also be very important. And in some cases, you can differentiate between species using this, or it may be even important to learn the number or observe rather the number of setae present on a particular area of a mosquito. Also on the thorax, we typically have the wings because they require the largest amount of musculature for movement. And the wings are vital to accurate identification. The placement and coloration of wing scales, veins, and bristles should be a primary focus when you go to learn species. As you can see in the lower left picture here, you see a grouping of dark scale patches, and then some places it looks like the scales are linear, and then they have a break in the linear line of scales that are dark, and then it switches to light. Those kinds of distinctions become very important when it comes to narrowing down your mosquito specimen to species. The presence of bristles, as you can see over on the right-hand side, the bottom right, is also very important. If it has bristles, if it doesn't have bristles, is what you typically would run into. But it may also contain where the bristles are located, or what the number of bristles is. In general, too, when you talk about insects, wing venation becomes extremely important. knowing where the wings veins are, where they bifurcate, how many there are. Each wing vein is often given a name, or in most cases in keys, they're given a numerical association, in which case you'll be looking to see if those associations from the key line up to the organism. Particularly if a lot of the other identifiers are knocked off, such as your scales. So because each of these wings is accompanied or is equipped with scales, that we often look for the patching or the breaking or the grouping of those scales. They can be knocked off during collection, and if they're knocked off during collection, venation becomes even more important for identification. All right, so in addition, on the thorax, you also have the legs. Special attention should be paid here to the banding or the lack of banding persistent across specimens. And when we talk about banding, we're mostly going to be referencing apical and basal banding, and in some cases cross banding. So knowing the distinction between apical and basal banding is probably the most important thing that we'll talk about in this entire presentation. Apical banding is banding before the joint. So if you look at the top section there where it says apical banding, you'll see the wider white band on the black leg just before the segmentation of the joint. All right, and basal banding means immediately following the joint. So underneath that, where you see the signifera specimen leg, you see the TA1 and the TA2 is distinctly divided by basal banding. And then you have cross banding, which may also exist where bands are both apical and basal. So if you have a band that stretches across the joint, that would be considered a cross band. Okay, and different keys use different language when it comes to describing the bands. But generally speaking, knowing apical and basal and their orientation within those legs, or on those legs rather, will get you the information that you need. If you look at the leg down in the bottom diagram, you can see an even clearer depiction of apical and basal as I've laid out here for easy reference. On the abdomen, abdominal shape is also important. Looking and distinguishing between a pointed and a blunt tip can be tricky, but it is vital in progressing through the identification key. In fact, knowing the difference between a pointed and a round or blunt tip is often one of the first identifiers you'll need to learn to progress through the genus key, where you go to narrow things down so you can switch over to the species identification. And you can see here in this particular slide that there's a distinction between pointed abdomens and blunt abdomens. And this should help you when you're looking under a microscope to tell which is which, because as much as we have experienced identifiers, oftentimes we'll still run into people who struggle with identifying between a round and a pointed tip. Okay, so furthermore, here as we move on in the abdominal identification characteristics, we again come to banding. Banding can be very distinctive in this section too, so knowing the difference between apical and basal banding is extremely important, because it'll allow you to help identify where the banding occurs on each abdominal segment, which would then can lead you to proper identification as you run through your key. With that all being said, this covers the basic mosquito biology, IPM structure, and some basic identification practices that should help get you started in your dichotomous key as you progress through it. If you guys have any questions, you can feel free to contact me directly if you wish. It doesn't matter what municipality you're from. I'm happy to answer questions at any time. You can just simply email me at rob106@Henrico, which is HENRICO.us. Thank you.